Intracellular iron homeostasis is regulated by ferritin, transferrin receptor, ferroportin, duodenal cytochrome B, divalent metal transporter 1, and other proteins. The translation of these proteins is regulated post-transcriptionally by the binding of iron regulatory proteins to three-dimensional stem loop structures located in the 3' or 5' untranslated region of the messenger RNA of these proteins. The stem loop structures are called iron-responsive elements. This mechanism for intracellular iron homeostasis is best understood for the translation of transferrin receptor and ferritin. In cells with sufficient iron levels, the iron regulatory proteins, or IRP1 proteins, contain iron-sulfur clusters with 4 iron atoms coordinated with 4 sulfur atoms. In this form, the iron regulatory proteins have cytosolic aconitase activity and are unable to bind to iron-responsive elements. During iron deficiency, the iron-sulfur clusters lose an iron atom, resulting in a 3-iron, 4-sulfur cluster. In this state, the iron-sulfur clusters dissociate from the IRP proteins. The IRP1 proteins are now able to bind to the multiple iron-responsive elements in the 3' untranslated region of the transferrin receptor messenger RNA. Binding causes the transferrin receptor messenger RNA to become stable and resistant to degradation. Therefore, during iron deficiency, translation of the transferrin receptor occurs, and iron uptake into the cell increases as a result. The IRP1 proteins also bind to the single iron-responsive element in the 5' untranslated region of ferritin messenger RNAs. Binding restricts the access of small ribosomal subunit binding to the 5' methylguanosine cap of the ferritin messenger RNA, resulting in the inhibition of its translation. Thus, iron storage is inhibited during iron deficiency.